Hello, everyone, and welcome to today's webinar, Breaking Through RT-qPCR Boundaries in Gene Expression Analysis. I am Matt Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. All questions submitted will be answered via email following the broadcast. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Jennifer Sandoval, Senior Manager of Molecular Biology at Thermo Fisher Scientific. Jennifer, you may now begin your presentation.
Thank you to all of you for joining today to discuss RT-qPCR and its importance in gene expression analysis. Before we get into the presentation, I would like to notify everyone that there will be polling questions that will appear throughout the talk today. As they appear, if you could please take a moment to answer the brief questions presented, it would be most appreciated. During this webinar, we will give a brief overview of reverse transcription and reverse transcription qPCR or RT-qPCR. Then we will discuss the attributes of reverse transcription enzymes that are critical for successful RT-qPCR experiments. The rest of the time will be dedicated to tips and tricks for overcoming RT-qPCR challenges, particularly the challenges that occur when working with very difficult RNA samples. Okay, so let's get started with the overview. Back to the basics of the different methods. As you know, reverse transcription is basically the synthesis of cDNA using RNA as a template. The figure here shown highlights the reverse transcription process in yellow to identify where it fits in the flow of genetic information. In nature, reverse transcription is involved in a number of processes in different organisms. The most well-known of the processes is the propagation of retroviruses. In fact, most of the reverse transcription enzymes that you use in the lab originated from these retroviruses. Besides those, reverse transcription also plays a major role in retrotransposon migration, as well as telomeric extension during cell proliferation in new carriers. Of course, this is also found in bacteria during synthesis of multi-copy single-stranded DNA. So really, a variety of processes in a variety of organisms are regulated by reverse transcription. Reverse transcription is also being utilized for many different types of applications. For example, in RT-qPCR, reverse transcription is combined with quantitative PCR to measure gene expression. In addition, RT-qPCR can be used to characterize different RNA species, such as transcript variants, long non-coding RNAs, microRNAs, and so on. RT-qPCR is also used to detect RNA viruses like influenza, Zika, and of course, coronaviruses. And in some instances, amplified qPCR fragments might also be used for downstream cloning applications and further analysis. For the purpose of today's talk, we will focus on the RT-qPCR workflow, which involves 5 main steps. First, we begin with our samples. These can come from a variety of sources, but for this purpose, let's say the samples are cells or tissues. Once the samples have been collected, the RNA from those samples must be isolated. After isolation, reverse transcription of RNA into cDNA occurs. The resulting cDNA is now the template for the qPCR step. At the end of this step, the resulting amount of amplified DNA has been quantified at every cycle, which correlates to the levels of gene expression. By design, the RT-qPCR technique is very sensitive, which is useful when attempting to detect very small differences in these levels, so that very small variants in gene expression can be detected. This enhanced sensitivity, however, also means that small variants introduced during the RT-qPCR setup or variants that are introduced during the workflow can have an effect on the RT-qPCR results, which can therefore result in variants and inaccurate gene expression data. In the initial phases of the workflow during sample collection and extraction, you might face a number of different issues like RNA degradation, low purity, and low yield, all of which affect RT-qPCR data. In reverse transcription, you might face a number of pitfalls. For instance, the RT reaction might not be efficient, accurate, and linear. This often depends on sensitivity, stability, and inhibitor tolerance of the RT enzyme used. Variation in pipetting of RT components can also contribute to inconsistency in RT-qPCR results. And as pointed out in this paper referenced here on the right hand of the screen, published by Bastian and the team, the quality of reverse transcription plays a significant role in achieving high accuracy for RT-qPCR results. Very commonly, poor and inaccurate RT-qPCR data actually arise due to a suboptimal setup. At the end, in the qPCR itself, there might also be issues, such as suboptimal primer design or outside sensitivity, as well as reaction inhibition. So in order to obtain sensitive and consistent RT-qPCR results, you need to avoid and or overcome these challenges throughout the experiment. In the webinar today, we will highlight ways to avoid some of these challenges. We will focus on the challenges that are related to executing reverse transcription and gene expression quantification with difficult RNA samples. Before we jump into the rest of the presentation, I wanted to first point out that, as I mentioned before, a common source of variance in the RT-qPCR workflow may arise simply due to issues with pipetting. For RT sets only, as you know, we have to pipette a number of different components: RNA templates, primers, we need, of course, dNTPs, buffers need to be added. RNA inhibitors are highly recommended to enclose the reaction and maintain integrity of the RNA template. Then we need water, and finally, reverse transcription enzymes. The point here is that there are many, many components of a reverse transcription reaction. If they're added individually into the reaction mix, errors could be introduced. So, to help minimize these errors, for RT-qPCR in particular, it is highly, highly recommended to use a master mix that contains all the cDNA synthesis reaction components in a pre-mixed format. Not only will this reduce the variation in your experiment, but it will also reduce the chance for contamination in addition to saving you time. Looks like it's time for our first polling question. Please take a moment to let us know what type of challenges you have already faced while performing RT-qPCR in your lab. Hopefully that gave you adequate time to answer. And now that that is out of the way, let's turn to important considerations when choosing reverse transcriptases. Not all reverse transcription enzymes are the same. These enzymes can vary in a number of different aspects, dependent upon things like RNase activity, thermostability, versatility, sensitivity, inhibitor tolerance, and just overall performance. Contrary to what many researchers may think, your choice of RT enzyme can have a strong impact on your RT-qPCR data in both quality and accuracy. Here's one example that demonstrates this aspect. The expression levels of 14 different genes were examined by an RT-qPCR assay using 11 different RT reagents. For each gene, RT-qPCR was performed with the same qPCR master mix, but using 11 different RT reagents. The resulting CT values were normalized against a CT value of 1 for the superscript 4 Velo master mix, which is shown in red. The other master mixes that were used all have a normalized full change of less than 1, meaning that they are less efficient and getting higher CT values than the superscript products. The thing to observe here is that there are huge differences in the data among the RT reagents. The efficiency of the master mixes is all over the spectrum, which means that the choice of RT reagents for your experiment has a huge impact on your CT value, which affects your end RT-qPCR results and ultimately your interpretation of your gene expression results. So given all of these variables, how do you choose the best reverse transcription reagent for your RT-qPCR application? It's always best to first consider the type of RNA sample you are working with, and then follow up with the aspects of your reverse transcription reagent. In the next couple of slides, we will discuss quickly these RT enzyme attributes and why they are important. First, let's take a look at the RNase H activity of reverse transcriptases. Natural or wild-type reverse transcriptases have RNase H activity, which will degrade RNA in the RNase cDNA hybrid. This is shown here in the top figure. When we need to synthesize long cDNA, this activity is detrimental because it can cleave off RNA too early and result in truncated cDNA. In addition, RNase H activity may reduce enzyme sufficiency because it can potentially compete with polymerase activity. In recent years, RNase H minus mutants have been engineered to reduce or completely remove RNase H activity. This helps to increase cDNA at length, but also it helps to increase overall robustness and efficiency of the enzyme. So if you have poorly expressed genes, degraded RNA, or low amount of RNA template, be sure to choose an RNase H minus enzyme in order to increase your cDNA yields for subsequent qPCR amplification. Another important aspect to consider is the ability of the enzyme to withstand high temperatures. This is also called enzyme thermostability. Typical wild-type enzymes work optimally at somewhere between 37 and 42 degrees Celsius. Thermostable RT enzymes can work at 50 degrees Celsius or higher. So why is that important? This is especially helpful when your RNA template contains very strong secondary structures or high GC content. These structures will hinder RT enzymes from binding to your RNA template, therefore reducing overall efficiency. Temperatures above 50 degrees Celsius are recommended in these situations to help facilitate the separation of these strong secondary structures of RNA molecules. Highly thermostable reverse transcriptases can tolerate these higher temperatures, and therefore using one can increase your cDNA yields and give lower CT values. Using thermostable enzymes with elevated temperatures can also help increase specificity when performing RT-qPCR with gene-specific primers. Another important reverse transcription enzyme property is processivity, which is defined as the number of nucleotides that are incorporated by an enzyme during a single binding event. On the left side of the image, we see an enzyme with very low processivity. It incorporates only a few nucleotides each time it binds to a template. These types of enzymes are slow and less efficient. Luckily, there are now engineered enzymes in the market that have been designed for higher levels of processivity, resulting in a higher number of nucleotides being incorporated during each binding event, as shown on the right side of the image. But why does this matter? When you are using an enzyme with a higher level of processivity, your reactions are faster. Beyond that, however, a high level of processivity increases the affinity of the enzyme template, therefore enabling the enzyme to produce a longer cDNA synthesis. So, the true added benefit of an enzyme with high processivity is its ability to read through RNA even when degraded or even when the RNA sample contains inhibitors. Since the enzyme is more robust, it's highly recommended to make this selection to ensure accurate and reliable RT-qPCR results when dealing with degraded or inhibitor-containing RNA samples. Now, as we mentioned before, RT enzymes also differ in sensitivity. This factor is especially important to keep in mind when studying gene expression of poorly expressed genes or when working with samples that have low RNA amounts. With low abundance or very low abundance targets, an RT reagent with poor efficiency will generate high CT values or no CT values at all. The blue curves on the graphs here represent a more efficient enzyme that has resulted in lower CT values for both high abundance and low abundance genes. thereby allowing and enabling more reliable quantifications. In order to detect RNA that is a low quantity, highly sensitive, reverse transcriptases are essential to ensure the accuracy of the quantification. Our final consideration is focused on inhibitor tolerance. Many scientists using RT reagents are often not aware that reverse transcriptase enzymes are highly susceptible to inhibition by many different compounds that can be found in the RNA preparation steps. Even after the RNA purification step, different compounds that can affect RT enzyme activity can still be present. For example, chemicals used in the RNA isolation process, inhibitors from biological sources, or simple reagents that are used for preservation of your samples can all be inhibitors of your RT enzyme. Some of these inhibitors are incredibly difficult to avoid and or remove, which means that they will be present in your RNA sample regardless of the purification method selected. Therefore, it is highly recommended to use an enzyme that is resistant to these inhibitors in order to get reliable RT-qPCR results. Later in this webinar, we will discuss these inhibitors at greater length and demonstrate the huge differences in the way that enzymes deal with these inhibitors, and then the effect that this has on your RT-qPCR results. So now that we have reviewed all of the different features and considerations of reverse transcriptase enzymes, let's discuss the best way to choose a reverse transcriptase enzyme for any given application. The table displayed here can help with the decision process. There are essentially three different types of reverse transcriptases: AMV, MMLV, and engineered MMLV. AMV RTs were one of the first reverse transcriptases to be used in the lab. This RT is relatively thermostable, but has very high RNase H activity, which limits its cDNA synthesis capability to 5 kilobases or less. Next up is the MLV enzyme. This has historically been a popular choice due to its reduced RNase H activity when compared to AMV RT, but with a target length synthesis ability of up to 7 kilobases. The last option, the engineered MMLV RT, has been designed for optimal cDNA synthesis. There are a large number of these engineered enzymes available, some of which can produce higher cDNA yields in shorter reaction times with a variety of sample types. Also, because these enzymes have reduced RNase H activity, high processivity, and very high thermostability, they are an excellent selection when working with any type of RNA sample to produce accurate and reliable RT-qPCR results. Selection of this type of enzyme is most critical, however, when working with challenging or suboptimal RNA templates in order to avoid potential failures of your reaction based on your enzyme selection. Some examples of this type of sample would be a subpopulation of cells, or maybe clinical research samples that have limited RNA amounts, working with a gene of interest that is expressed at a very low level, genes that are high in GC content or secondary structure, or when working with samples where RNA integrity has not been adequately preserved, like animal tissues, blood, and plant samples, or samples that have high presence of inhibitors such as soil and FFPE samples. In all of these cases, an engineered MMLV RT would be the recommended enzyme of choice for sensitivity and efficiency. In the coming slides, I will show some RT-qPCR data when working with these challenging samples and discuss how highly efficient reverse transcription reagents can help you to achieve high quality data. Before we get into that, let's first discuss specific ways to overcome RT-qPCR challenges when working with difficult RNA samples. But first, time for another polling question. Please let us know if you would like to be contacted by our team to help in your selection of products. Okay, moving on. Many reverse transcription reagents can effectively transcribe highly abundant RNA targets. When analyzing gene expression from limited sample sources, or when analyzing expression of low abundant genes, however, these enzymes often fall short. If you are working with these challenging types of samples, it is highly recommended to check both the sensitivity and dynamic range of your RT reagent and RT-qPCR reactions. Here we have an example of results from a high abundance gene, GAPDH, and a low abundance gene, POLE, using an RT-qPCR assay with two different RT master mixes. In both cases, we have a linear dynamic range spanning from 1 picogram to 2.5 micrograms of total RNA. Both master mixes use superscript 4 velo and superscript velo, provide detection of RNA down to 1 picogram for both genes, which demonstrates that they are both efficient, sensitive, and appropriate selections for both high and low abundance gene. The superscript 4 velo master mix, however, that's shown in red, does have lower CT values, meaning that it is more efficient and provides higher cDNA yields with the same high linearity across the widest range of RNA inputs. If you are working with small RNA quantities, make sure to test the dynamic range of your enzymes and ensure that your setup will effectively work with your low input RNA sample. When your RNA template is degraded or it contains inhibitors, selecting RT reagents that are highly sensitive and efficient can make a huge difference. In the example shown here, we used RNA isolated from whole blood, which had a large amount of degradation in the sample. When we compared RT-qPCR data from this sample using 5 different cDNA synthesis reagents, we found that all of the reagents used were able to transcribe RNA from the high abundant gene GAPDH, which is shown on the left-hand side. On the right-hand side, however, only one of the 5 reagents was able to transcribe the RNA for POLE, a low abundance gene, from the limited RNA input sample. As such, we can conclude that a highly efficient reverse transcriptase is critical for the detection of low abundance targets when working with highly degraded samples in which your RNA input will be at lower levels. If you are working with these types of limited samples, such as isolated circulating cells, stem cells, or clinical or research samples, you know that RNA isolation might not be possible since this process can and often does result in the loss of RNA. So what can you do? One possible solution is to use specialized reagents that allow you to perform reverse transcription without RNA purification, such as the Invitrogen Cells Direct cDNA Synthesis Kit. With this kit, you can go from cell collection, culture, and mammalian cell lysate directly into cDNA synthesis in a single tube. Since RNA isolation and purification is not necessary, loss of your total RNA sample is prevented, which is ideal when working with low RNA input. Another option for low input RNA samples is the Invitrogen Superscript 4 single cell low input cDNA preamp kit. This kit provides direct cDNA synthesis and amplification from intact single cells. The single tube protocol is ideal when trying to utilize minimal bias while increasing the number of measured targets from a single sample. Because the kit contains the superscript 4 RT enzyme, it maintains high levels of sensitivity, full-length cDNA yields, and is also compatible with downstream analysis by NGS or qPCR. Okay, now let's discuss another RNA sample challenge, RNA with high structural complexity. As previously stated, reverse transcription has a tendency to be less efficient when working with RNA that is rich in secondary structures. These structures can hinder your enzyme from reading through the RNA template, thus resulting in lower yield and inconsistent CT values. Your inclination may be to run your reverse transcription reaction at a higher temperature to release the secondary structure. Makes sense. Be careful, however, when doing this, because elevated temperatures can often denature reverse transcriptases, especially wild-type natural enzymes such as AMV or MMLV RTs. you can see the need for using RT enzymes with high thermostability in these cases. The data shown here compares reverse transcription reactions using thermostable versus non-thermostable enzymes. As you can see, at about 55 degrees, reverse transcription efficiency is drastically reduced when using non-thermostable enzymes, as seen from the fainter and missing bands on the right-hand gel. Now, for this kind of RNA, it is also desirable to have highly processive enzymes, so reactions can be carried out at higher temperatures in a shorter time. In the figure on the right, reverse transcription using different master mixes was performed at various specified temperatures for 10 minutes. The red circles show results from using the superscript 4 Velo master mix, which contains a thermostable and highly processive RT enzyme, which produced more comparable cDNA yields and CT values across the range of temperatures tested. Next, let's discuss challenges when working with RNA from tissue samples. Whether you are working with fresh or archive samples, challenges may arise simply due to the inherent nature of these tissues or due to the way the sample was processed. In the following slides, I will review helpful tips and tricks when working with RNA from fresh, frozen, FFPE, and archived tissue samples. RNase tissue can easily become degraded during sample collection and handling. As such, the question arises as to help preserve the RNA during tissue handling. One reagent that we have seen to be helpful in this situation is the Invitrogen RNA Later solution. RNA Later solution is a tissue storage reagent that rapidly permeates tissue to stabilize and protect cellular RNA. A bonus is that it's very easy to use. Fresh tissue samples or cells are first added to the RNA Later solution, where RNA can be stored for up to 1 day at 37 degrees Celsius, 1 week at room temp, or indefinitely at minus 20 degrees Celsius. When you are ready to proceed with your experiment, simply remove the RNA Later solution and continue with your extraction procedure. The data shown on the right-hand side of the slide shows the integrity of RNA isolated from a variety of tissue types and stored in the RNA Later solution for different durations and at different temperatures. As you can see, all of the samples have successfully preserved the RNA under all conditions. This approach is highly recommended in circumstances where your RNA is at risk of degradation during the sampling process. There are some instances where complete protection from RNA degradation is just unavoidable. In these cases, it's always recommended to use a highly processive and sensitive RT enzyme to achieve accurate and reliable results. As an example, here we have shown RT-qPCR results using degraded RNA from frozen lung tissue samples. The RIN for all of these samples was less than 5, so fairly degraded RNA. CT values, as before, were normalized to the superscript 4 Velo master mix. Across the board, the different RT reagents used resulted in very different efficiencies with this degraded sample type. This is highlighted just to again stress the importance of your RT reagent choice in order to obtain high-quality results. Okay. Now let's take a look at challenges that are associated with FFPE samples, where RNA often gets degraded due to the way in which the samples are processed, both during tissue preservation and also retrieval from the blocks, not to mention the additional complications of low RNA yield and inhibitors that are concerned with FFPE sections. To overcome these challenges, make sure to choose an isolation method or kit that has been specifically designed for FFPE samples. There are a number of different kits to select from, and the table shown here highlights the critical components of each option. Each type of kit differs in things such as expected yields, preparation time, the overall process, and compatibilities. Selection of the appropriate kit for your application can help to overcome the hurdles experienced with these FFPE samples. In this example, a number of different RT reagents were used for RT-qPCR on FFPE samples. You can clearly see that there is a large range of differences in the results across 3 genes, B2M, GAPDH, and ACTB. The RT enzyme with a high sensitivity to detect low amounts of RNA, a high processivity to transcribe fragmented or degraded RNA, and a high inhibitor tolerance to withstand inhibition from carryover chemicals, which just happens to be superscript 4 velo, it far outperforms the other RT reagents used, as you can see in the red bars on the graph. Let's move on to working with RNA that is isolated from blood samples. RNA from blood samples is often degraded due to the elevated amounts of RNase that are naturally present in blood. The image shown on this slide is looking at RNA that is extracted from cultured cells on the left compared to RNA from whole blood samples on the right. As you can see, the RNA from whole blood is heavily degraded and presents a common set of issues when dealing with this type of sample. In addition to the degradation, RNA yield may also be low depending on the amount of blood cells that are available in the sample for RNA extraction. Inhibitors are also a factor when working with whole blood, mainly due to the heme anticoagulants or heparin that may be present in the RNA. There are a lot of challenges to overcome when analyzing gene expression from whole blood RNA. One way to overcome blood RNA degradation is to collect the samples using RNA stabilization reagents. These reagents can lyse blood cells while inactivating RNases that are present during precipitation of the RNA, all of which are important steps to prevent degradation. One such type of reagent is the Applied Biosystems Tempus Blood RNA tube. When using this product, blood is collected in the tube and mixed with the stabilization reagents to create a stable RNA sample that can be stored up to 5 days at room temp, up to 7 days at 4 degrees Celsius, or for longer storage at minus 20 degrees Celsius. However, in many instances, such as when you are using clinical blood samples for research, collecting blood in RNA stabilization reagents might not be possible. When this is the case, it is critical to select a robust enzyme that can handle the low input and degradation that is present in your samples. Shown again with this example, our highly effective superscript 4 velo master mix, which is highlighted in the dashed line ovals, is making a difference. This enzyme is generating not only lower CT values, but also less variation. Finally, when working with blood samples, inhibitors and anticoagulants are always going to be a factor. They are typically present in blood RNA even after RNA purification using a variety of methods. And as no surprise, different RT enzymes are going to have different susceptibility to those inhibitors. Here we show how heparin and hemin can inhibit your RT-qPCR experiments. As you can see, some of the RT reagents used are more resistant to inhibition than others, analyzing on both whole blood samples as well as the inhibitors themselves. Just a reminder, please select carefully when deciding on a reverse transcription reagent to use with these samples. We know that analyzing gene expression from animal and human tissues can be a challenge, but with the advances in robust reverse transcriptases that are available, reliable results are achievable even when working with challenging sample types. So let's now turn our focus to working with a different template, plant samples. Plant samples pose unique challenges in that different types of tissues, plant developmental stages, variety of species, and so on may require different RNA isolation techniques. In addition, plant compounds like polysaccharides, phenolics, and flavonoids have been shown to inhibit reverse transcription enzymes. Finally, poor RNA integrity is a common issue occurring after extraction from plant samples. The gel shown here is demonstrating the high levels of RNA degradation that is commonly observed across a wide variety of plant tissues. When plant RNA is reversely transcribed with different reverse transcription reagents, large differences in CT values and differences in RT-qPCR results are observed. In this example, we have tested 7 different reverse transcription reagents with 3 different plant RNA sources to detect 6 different gene expression levels. No surprise again. There is a large variability in the end results. Also, considering the effects of inhibitors within these plant-direct extracts, we can clearly see that some enzymes are more resistant than others. In addition, you can see here from the lower CT values that some RT enzymes are more efficient than others when working with degraded RNA in these plant samples. The more robust RT reagents also provided more consistent and reliable data, as shown by the lowered standard deviation, which should provide confidence in your RT-qPCR data when using these enzymes. Okay, one more polling question before we wrap things up. Please let us know if you would like to be contacted for quote assistance. We appreciate your participation. I hope you found all of those tips and tricks beneficial for RT-qPCR analysis for animal, human, and plant tissue RNA samples. For our final consideration, let's take a look at one final common issue that occurs with the RT-qPCR workflow across almost all of these sample types: genomic DNA contamination. Common issues that can arise due to genomic DNA contamination of isolated RNA include false positives, which lead to inaccurate gene expression results. Trace amounts of genomic DNA are very commonly co-purified with RNA, even when using the most optimized RNA isolation techniques. This gel image is an example of what genomic DNA contamination might look like, with the upper band being detected in lane 4. Although we can see it here, please also keep in mind that in a majority of cases, genomic DNA might not be visible on the gel due to its low quantity present in the sample. Nevertheless, since RT-qPCR is a very sensitive technique, even small traces of genomic DNA can lead to false positive RT-qPCR results, leading to inaccurate quantification of gene expression, especially if you are quantifying low abundance genes. A good solution is to use a control reaction without any reverse transcriptase to detect any genomic DNA that might be present. This is shown in green on the graph. Here we see amplification even without an RT enzyme, which clearly indicates contamination with genomic DNA. Another solution is to use exon-exon spanning primers in qPCR that are designed such that genomic DNA containing the intron cannot be amplified. In this case, if you have genomic DNA contamination, it will not be amplified in your sample, thus separating out the RNA from the DNA. A final option is to treat the RNA sample with DNase 1 to remove the genomic DNA. However, although DNase 1 cleaves double-stranded DNA, which is the genomic DNA, it can also cleave single-stranded DNA, including your primers or cDNA. So, in order to preserve the integrity of your reaction, DNase 1 must be completely inactivated and removed prior to the reverse transcription step. This inactivation is usually done by adding EDTA and heating at 65 degrees Celsius. This step, however, is quite harsh for your RNA and might lead to RNA degradation. Since this can have a negative impact on your RT-qPCR results, especially when working with low input RNA samples or poorly expressed genes, you might want to consider using a modified DNase reagent like DNase. Invitrogen DNase enzyme has been designed to be specific for double-stranded DNA cleavage. As such, it will not degrade your single-stranded primers or cDNA when in the reverse transcription reaction. DNase can be easily deactivated at 55 degrees Celsius for 2 minutes if desired, but this step is not necessary due to the enzyme's double-stranded specificity. If you choose to complete the inactivation step, however, because it is performed at a lower temperature and for a short time, the integrity of the RNA is less impacted. The graph here highlights RT-qPCR data on RNA that has been treated with either easy DNase or with DNase 1 in the mandatory inactivation. The DNase 1 treatment and inactivation, represented by the dark gray dots, resulted in increased CT values when compared to the easy DNase, represented by the red dots, in the RT-qPCR assay. That suggests that there was some RNA degradation present in the DNase 1 treated sample that was not present in the easy DNase treated sample. Using easy DNase to control your genomic DNA contamination can really help to streamline and easily preserve the integrity of your RNA preparation before you perform RT-qPCR. So, just to recap, traditional reverse transcription workflows using DNase 1 are time-consuming, have removal steps that can degrade your RNA, and often fail to remove gDNA completely, which leads to inaccurate results. Easy DNase can be provided in a single tube with your superscript 4 one-step RT-PCR system or the superscript 4 Velo Master Mix for a simple one-tube workflow. Wait, did you catch that? A single tube is used. Then a gentle 2-minute gDNA removal step and an RT reaction in just 10 minutes, bringing your total time to 27 minutes versus 105 minutes with the traditional method. Very streamlined and completely a time saver for this part of your experiment. So in research these days, we're using more and more relevant but technically challenging samples, such as blood samples, tissue samples, biopsies, and others, which often yield low input, degraded, and/or inhibitor-containing RNA. One of the biggest challenges with those sample types is how to get highly reliable gene expression data. In all RT-qPCR assays, but especially in these circumstances, reverse transcription is a critical step that needs to be carefully optimized to ensure reliable and accurate gene expression interpretation. As we highlighted today, not all RT enzymes are the same. Differences in efficiency and reliable performance become even more exaggerated when working with suboptimal RNA preparations. RT enzyme properties such as RNase H activity, thermostability, processivity, sensitivity, and inhibitor tolerance can and will impact the success of reverse transcription and therefore have to be carefully considered in your RT enzyme selection. Challenges within RNA samples can range from low input, degradation, inhibitors, and secondary structures. And finally, to conclude, keep in mind that only highly efficient reverse transcriptases have the ability to overcome these challenges to generate high yields, low variability, and faster reaction times for all types of RNA samples. Thank you so much for your attention today. I hope that you found this webinar beneficial and that you learned some considerations to keep in mind to help you with your RT-qPCR experiments. To learn more about the data and or products you have seen today, please go to the links provided here for more information. If you are interested in more reverse transcription basics, considerations, or additional webinars, please visit our educational and webinar hubs listed. And lastly, you may also contact our technical support team directly using the Contact Us information URL that is listed. Thank you for your time and your attention.
Thank you, Jennifer, for your time today and your presentation. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share this email with your colleagues who may have missed today's live event. Until next time, goodbye.